Allosteric enzymes often do not show typical Michaelis-Menten kinetics because they, option A, do not reach steady state, option B, have two conformational forms with different activities, option C, lose activity during the reaction assay, or option D, have no enzyme substrate complex. So the correct answer would be option B. Allosteric enzymes often have multiple conformational states, typical an active form and an inactive or less active form. The binding of substrate or allosteric effectors can shift the equilibrium between these two conformations, leading to non-Michaelis-Menten kinetics. This results in a sigmoidal kinetics often observed in allosteric enzymes rather than a hyperbolic curve predicted by the Michaelis-Menten model.